cell and the nucleus of the cell with its important genetic information. Now we know that the genetic information is made of DNA. We also know that DNA contains a specific code. This code is in the sequence of the bases, the nitrogenous bases. And today we are going to see how that code on the DNA is used to make proteins. Protein synthesis comes with the expression of the gene. The sequence of the nitrogen bases will eventually tell us the code of the protein. I would love us to quickly look at proteins. Yes, we know that proteins are made up of a specific sequence of amino acids bound by peptide bonds. This sequence will then be folded into the secondary structure of a protein, which is held together by hydrogen bonds. Then we have the third or the tertiary structure, which make these bonds into a three-dimensional structure. Now this three-dimensional structure is held together by disulfide bonds and also hydrogen bonds. That is the tertiary structure. But proteins also have a quaternary structure. We have different polypeptides making proteins bond together. We need proteins because it makes up living material. We know blood contains hemoglobin. The brain needs the channel proteins in order to get the brain signaling small molecules to enter the nerve cells. We know at the synapses, this channels is important for calcium and sodium ions to enter. Enzymes need to digest inside the mouth, inside the stomach, the small intestine. But enzymes will be all over in the mitochondria. Enzymes will catalyze reactions, all made of proteins. Then we have clusters of proteins to help the cell copying genes and making cell division and new protein synthesis possible. Antibodies, proteins that helps with the immune system. We call them immunoglobulins. Cell messengers, the receptors at the cell surface membrane that needs to communicate with the inside of the cell. And also hormones are messengers, chemical messengers made out of proteins. Then we have the muscles and the hair and nails. We also divide proteins into globular and fibrous proteins. And just to help us to remember, let's add hemoglobin, globular protein. So it's a spherical shape. The channel proteins, globular. Enzymes, globular protein. Clusters, globular protein. Antibodies or immunoglobulins, globular proteins. The hormones and the receptor proteins outside the cell membrane, globular proteins. And then we have the fibrous proteins, the keratin in the hair and nails, and the hooves, feathers of other organisms, other animals, and then the muscle proteins are also fibrous proteins. Wow, now we did a lot of protein revision. That is the recipe, dear grade 12s. Repeat, repeat, repeat. Now, we are going to see how these proteins can be synthesized. So, we have a sequence of amino acids to be connected by a peptide bond to one another, and eventually it forms a polypeptide. The polypeptide is folded and bonded together to make a protein molecule. We have 20 different amino acids, and how do we get the amino acids? We get the amino acids by what we eat, right? So, this amino acids will be part of the ribosomes. The ribosomes will take all these different amino acids that we got from what we have eaten, the proteins that we have been eaten, will be digested, and I will have lots of amino acids in my blood. Now the ribosomes will have the job to assemble them into the correct proteins. Where do I find ribosomes? On the rough endoplasmic reticulum, and also lying free in the cytoplasm. If now I am looking at the functional components of protein synthesis, we of course will start with DNA, the double helix with a specific sequence of nitrogenous bases. This nitrogenous bases for the DNA, cytosine, that will always bond with guanine, and adenine, that will bond with thymine. Always bonded like that. It is not a random sequence, but this sequence will hold the key. This will be the code for a specific amino acid. 3 of them. Okay. Then we know that we have RNA, and especially the mRNA needs to link with DNA at a specific stage. So it also have cytosine, which will link with guanine when it needs to link with the DNA. Adenine will now link to uracil. Then we have the RNA. So here we have our DNA molecule. DNA contains cytosine and guanine. Guanine and cytosine are linked in this double helix. We call them complementary strings. They do not look the same. On the one side, if I have cytosine, on the other hand, I will not have cytosine but guanine. Then I have adenine and thymine, which will link together. Okay, then I have an RNA strand, which is a single strand. And here I see, okay, cytosine and guanine resembles DNA, adenine resembles DNA, but uracil is a different nitrogenous base or nucleobase that I find on RNA. So why? To make protein, we need the following to take place. We need transcription. So, transcription means to copy the genetic information from DNA to RNA. We call that transcription. RNA will copy the genetic information. But the copy on the RNA will be the complement of the DNA message. You agree? That we call transcription. Why do we have transcription from DNA? Because DNA contains the genetic code for protein. It is needed to get that message to the ribosomes that is outside the nucleus. But the DNA is large and the double helix cannot leave the nucleus. So therefore, what needs to happen is the DNA needs to unwind, unzip, and to unzip it needs breaking the hydrogen bonds between the nitrogen bases. Now the messenger DNA will come and make a copy. Again, it is not an exact copy, but it will form the complement. It will form the complement of the message. Because the single strand of RNA can leave the nucleus through the nuclear pores. So what will happen is, if we can just go back to our previous slide, the guanine of RNA will link to cytosine. So let's make a note here and to help us to remember. The guanine on the RNA will link to the cytosine on the DNA. Right? There we have RNA, guanine will copy the cytosine. The adenine of the RNA will link to the thymine of the DNA. The uracil on the RNA will link to the adenine. Right. The cytosine on the RNA will link to the guanine on the DNA. I hope you do understand that. So now we have a transcription, a copy of the DNA that opened to have the part with the specific code for a specific protein open. So, transcription, a part of the DNA unzips, unwind, unzip, breaking the hydrogen bonds. mRNA comes and take only the part with the code on and copy the code from the DNA, but in a complementary message. It will copy the complement and there will be no thymine but uracil. That is what we call transcription. How does it take place? There are free RNA nucleotides inside the nucleus, and that was produced by the nucleolus. Make the RNA, the free copies attach in the correct complementary order, and now it is called transcription, leaving the nucleus. The mRNA goes through the pores with the message. It's taking this message to the ribosomes. In the ribosomes, there are transfer RNA. Transfer RNA, which is, this is a tRNA or a transfer RNA. We see that attached to each transfer RNA is an amino acid. So this amino acid will only be attached to this specific 3 bases. This 3 bases is called the anticodon. This anticodon inside the ribosome will fit 3 specific sequence of a mRNA, and that we call a codon. On the RNA, mRNA, we have a codon. And just to help us to remember that it is 333, they call the 3 there that will belongs together to form a codon to fit a specific anticodon. On the DNA, they say there must be a triplet of codes on the DNA that will form the codon of the mRNA. On the codon of the mRNA, there will be an anticodon. Attached to that anticodon, we have the specific amino acid. And now we call this translation. When the codon is fitted out with an anticodon, now there will be a specific order of amino acids linked together, forming a peptide bond, and that is translation. So, let's quickly talk about translation again. The decoding of the mRNA, 3 bases of mRNA, will attach to a tRNA. The tRNA is like hooking 3 specific nitrogenous bases, because this 3 had that amino acid attached to it. As this one is leaving, a next one can attach in the ribosome. And there I will then have a specific sequence of amino acids which will form a polypeptide string. That is translation. The mRNA coding for a specific tRNA with its attached amino acid that will be sequenced inside the ribosome. That will be called translation. The transfer tRNA carries amino acid from the cytoplasm to the ribosome. It will attach to a specific 3. These specific 3 will be called a codon of mRNA attached to an anticodon on the tRNA. So where did this amino acids came from that is now in the cytoplasm? That came from the proteins that I ate. It got digested or hydrolyzed inside my digestive system, absorbed by the blood, transported to my cells, and now the amino acids can get into the cells. This tRNA, together with the code on the mRNA, rearrange all the amino acid so that I can have a new protein with a specific function in the human or any other animal or plant cell. And again, a series of 3 adjacent bases on the mRNA is a codon, and a series of 3 nucleotides that are complementary to the codon will be on the tRNA. What will be the complement of this codon? G will have a C, C will have a G, A will have a U, because we are talking, and this will all be attaching to a specific amino acid on the tRNA. Right, so each tRNA codes for a different amino acid. Messenger RNA do transcription. mRNA is transcribed in the nucleus. The mRNA can leave the nucleus through the nucleopores because it is small enough. It moves through the cytoplasm and joins the ribosomes. Inside the ribosomes, which is the factory of proteins, the codon of the mRNA will be translated by tRNA. The mRNA enters the cytoplasm attached to a ribosome. Translation begins at AUG, which is a start codon. Each transfer RNA, tRNA, has an anticodon whose bases are complementary to a codon onto the mRNA strand. The ribosome positions the start codon to attract its anticodon, which is part of the tRNA, which binds methionine. The ribosome also binds to the next codon and its anticodon. And then I have an assembly line of amino acids joined by a peptide bond to form a polypeptide. When I have a polypeptide and it completes its 3-dimensional structure, I have a protein. So we can use a codon chart, which is fortunately set by all the clever scientists that researched protein synthesis, and now we can discover which amino acids will fit. So when I have CAC, I look for C in the first base, A, and here I have a C, right? Third base. So it will be histidine. You agree? You can pause the video and see if you can find another one yourself. C. Again, C. The second one is C and an A. C, 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 A. There it is. So it will be proline. U. Let me use another color so that we do not get confused. U, G, U, G, A, U, G, A. UGG, first UGG. UGG is tryptophan, and then I have a stop in UGA. So, you got that? Right, then the next one is a different type of chart. to help us, and we start in the middle. A-U-G. Aha. So the first one will be methionine. Again, you can try and find them on your own, and then play the video again to see. There we see methionine, asparagine, aspartic acid, and then a stop. To capture this, we have DNA needs transcription by the mRNA to get the code of the gene to the mRNA. mRNA will be translated by tRNA, and that will form the protein. Add your own sounds. RNA polymerase, doing the unzipping. RNA subunits produced by nucleolus. So I added a quick revision sheet for you so that you can quickly see and bind the correct names in your pathways in your brain, dear class. Please print this one as well as the added summary that also will help you to quickly revise it and the worksheet attached. Please try your utmost best. I think that you then repeated it enough, and if there are any more things that is not clear to you, just watch it again or read through your textbook.
Hi there, parents. Do you have primary school kids and want them to get extra quality supplementary lessons to up their grades? Then simply switch to One Africa TV's #LearnOn1, where your kids can get maths, English and science lessons every weekday from 9 to 11 a.m. with repeats from 2 to 4 p.m. And thanks to NMH's MyZone, these lessons are 100% Namibian. There's so much to learn with #LearnOn1. Invite learning in.
and welcome back, grade 12s. It is so good to know that you are all listening and you are all prepared to have a lesson in biology. Remember, biology, the spice of life, you will need it for your existence, for your life, for the rest of your life. Please listen to this lesson as if you are preparing yourself not only for life, but definitely for your exam at the end of the term and at the end of the year. So we are going to look at chapter 29, and we are using our textbook that was done by Appel de Klerk. Inheritance, and specifically DNA and chromosomes. You will see we are going to do a lot of terminology today. It will be a good thing to make sure you know the definitions. So, my first assignment to you is complete this worksheet. Write down the title, draw the diagram, and see if you can get the labels. Please pause your video and complete it. If you then are ready to continue and reach the answers, you can continue to play. Okay, so I hope that you remember we always have a heading for a diagram. Remember, we use at least half an A4 page, or when they give you a block or a square, we fill it with our diagram, and our labels must also fit into that space. We draw in pencil. Labels and label lines are in, yes, you know that, in pen. We never cross our label lines, and we do it as neatly as possible. We cannot use cursive writing with a diagram. So we have the endoplasmic reticulum, the nucleolus, chromatin, nucleoplasm, nuclear pore, and the nuclear envelope. I want you now to see. Remember, we need to know and do revision. What is revision? To repeat. And that is how we study. So, I want you to copy these labels and then see if you can find the function. Yes, you will have to go to the start of your textbook to find these functions of all these parts. Pause the video and complete it first. Then you check, do you know them? Okay, so the nucleus has the function to control all the functions and activities of the cell. It passes on genetic information to new cells, and it is the site for transcription. And transcription is where the recipe of the proteins are being shared. We will come to that process in the next few lessons. The nucleolus is making the ribosome synthesis, and it contains the RNA, which is also a type of nucleic acid. RNA from the nucleolus. Then we have the rough endoplasmic reticulum. It transports a specific RNA, the messenger RNA, to the ribosomes. Ribosomes is the site where protein synthesis, the building of proteins, will take place. What is the chromatin? Chromatin is a long thread that is made up of DNA, deoxyribonucleic acid. So it's something else than RNA. RNA in the nucleolus. DNA is the building material of the chromatin, the long thread full of genetic material in the nucleus. The nuclear envelope contains pores, and that pores will be the outlet for the RNA to the ribosomes to make proteins. We have the nuclear envelope that makes the boundary of the nucleus, and we have the nuclear pores that we call the openings. So let's see, what is a chromosome? A long thread-like structure in the nucleus that carry the genetic material, and it is composed of DNA. So a chromosome is then that long chromatin network that will come shortened and making a short fat structure. And if you can look here, you can see it is actually rolled up very tightly to make a chromosome. Now, this is called a chromosome. But a chromosome actually consists out of two chromatids. This contains genetic material, this one contains genetic material, and it is then replicated so that the other chromatid has exactly the same genetic material. If I just can explain again, we have the chromatin network, a long thread-like structure that is lying in the nucleus. This is for the phase of the cell where the cell is doing its function. But when this cell wants to divide, the chromatin network will shorten and form short, fat chromosomes. A chromosome consists out of two chromatids, one chromatid and the other one, they are replications of exactly the same material, so that I actually have two parts with exactly the same genetic code on it. It is just kept together by the centromere to know that these two, this one and this one, it contains exactly the same genetic material. So the definition of a chromosome, long thread-like structure in the nucleus, carry genetic material made of, composed of DNA. So if I can look at a chromosome, here is a real photograph. Most of the time, the chromosomes are too thin to be seen except with an electron microscope, when it is that long thread-like chromatin structure. But when the cell starts to divide, they will become shorter, fatter, and very tightly curled up. And strings that are exactly the same are kept together by the centrosome. So if we look at the nucleic acids, we know that the nucleic acids, DNA, is the one that makes up the chromosomes. It is the one that we find in the nucleus, nucleoplasm. But They call it nucleic acids. At first, they only thought it is only found in the nucleus. But later they found that this nucleic acids can be at other places also in the cell. So we have the DNA, deoxyribonucleic acid, and ribonucleic acid. And yes, Joshua, I know that you will ask me now, do I have to know these names? Yes, make sure. Quickly scribble the names down on your notepad so that you can remember them and can write them down. So if I look at the chromosomes and I can unravel a chromosome, I will see the following. I will see that it is actually tightly curled up and packed string. This string is curled around proteins. You know, if I do not want my thread or my wool to get all knotted up, I will use something to roll it on. So this histones are proteins that is used to roll this thread around so that it is not getting a big confusion. And then as we increase the size, we get to the DNA's structure. And what we can see is we see a spiral. It is in a spiral shape and it looks like a ladder. And we will look at the DNA and it will look like this. It is spiral shape and it looks like a ladder. And for that we have a name, a double, because the ladder has two legs. It's a double helix. That is how we describe the shape of DNA, a double helix. If we now can look at the links between the backbone of the DNA, we see that these links are color-coded and they are a specific molecule. A and T are linked. Let's say blue and yellow is A and T. There I see blue and yellow. So blue and yellow, A and T always link, and G and C always link. And that G, A, and T and C will make a specific code. And that specific code made up out of G's, A's, T's, and C's is a gene. And we know that genes carry a specific code to make a specific protein. So, on the DNA, there are specific sequences of the G, A, C's, and T's. This specific code I called a gene. And a gene will tell me the recipe for a specific protein in the body. So, if we have our chromosomes, there's something else about chromosomes that we need to know. I have my chromosome with one chromatid. Then, when the cell wants to divide, when it's time for the cell to divide, which is controlled by the nucleus, the chromosome will double, and I will now see two chromatids, and they will be linked together by a centromere. But in my cell, I have genetic material that came from my father paternal chromosomes. But also, I have the same from my mother. And these two chromosomes will carry exactly the same traits. So with trait, I mean, if on this chromosome I have my recipe for my hair color, my strength of my muscles, and the shape of my ear. There I will have hair color, strength of muscle, shape of ear. There I will have hair color, strength of muscle, shape of ear. But what the codes that I got from my father. And that is, each one is called a gene. On this side, I also inherit from my mother hair color, but it is now doubled, muscle strength, and the shape of my ear. Yes, and when I have, when I have these two, they find each other and they will form a homologous pair. So, chromosomes from my mother and chromosomes from my father with the same human characteristics on each will come, they will search each other up, and they will form a homologous pair. So, each chromosome contains one very long molecule of DNA. The DNA molecule carries a code that instructs the cell about which kind of protein it should make. Each chromosome carries these instructions for making many different proteins. So a gene is, each chromosome is made up of a large number of genes coded for the formation of different proteins, which gives us our characteristics. A gene is responsible for a particular characteristic, and it is always on the same position on the chromosome. And that is how they now can find the gene that cause specific diseases or problems in the human body, because they know where the gene is to make the mucus inside your lungs. If that mucus is not made to a specific recipe and it's thick and sticky, you can suffer from a disease called cystic fibrosis. And that cystic fibrosis is due to a gene on the chromosome that do not know the proper recipe to make the good mucus that we need in our lungs. That's just to help you to understand. Here we have a plant example. The gene to tell us the color of the flower is on this chromosome. Okay, so this is one part of a chromosome. We know it is a chromatid, and this specific spot is for purple flowers, the recipe to make purple flowers. So because it is on this specific spot, we say the locus, the position, the locus for the gene to make purple flowers is here. Of course, this flower also inherits from his other parent a gene for the color of the flowers, but it has the gene for white flowers. Now I use the word allele. The allele for the flower color is on the one chromosome, purple flowers, exactly on that same locus in the homologous pair, I will find the allele for the color of the flower from the other parent. And in this case, it is white. And I hope that you now want to know, okay, if this nucleus has an allele for purple flowers and for white flowers, what will be the color of the flowers? Just wait and see. We will reveal this in a next lesson. What I want you to know is to understand that on the same spot, on the same locus, I find the genes to tell me color of the flower, and I call those two genes the alleles for color of the flowers. What we also know is that if I only have half the number of the chromosomes, I have the haploid number. So if I have only one copy of the genetic material, I only have one copy, I do not have this chromosome's homologous partner. I have only one copy of the genetic material, as what we will find in the gametes. I have the haploid number, and we use the symbol N. If I have two copies, so I have the homologous pair, both from the mother and the father. I have two copies of genetic material, which I will find in all somatic cells with a nucleus. I have the homologous pairs. And now I have a diploid nucleus. So please, class, revise and know these definitions. It's from page 481 to page 487, I think, and I want you to make sure that you know all the definitions before we start with our next lesson, where we are going to discuss the structure of DNA and RNA. Thank you very much.
Hey kids, are you in primary school and want to get extra supplementary maths, English and science lessons? Then invite Learning In with One Africa TV's #LearnOn1 every weekday from 9 to 11 a.m. with repeats from 2 to 4 p.m. And thanks to NMH's MyZone, these lessons are 100% Namibian. There's so much to learn with #LearnOn1. Invite Learning In.
Humanitarian crises are all too familiar to television viewers. Civil wars that cause huge movements of refugees, worldwide pandemics, and natural disasters like earthquakes and tsunamis. This is the way most of us are used to seeing these heart-wrenching events, on a screen in front of us, watching passively. But what if you could step into the frame and actually feel what it's like for the individuals on the ground?
People come out of it feeling enlightened and often moved and often ready to take action. A lot of people automatically say, well, what can I do? How can I get involved?
Garbo Aurora is leading a team at the United Nations who are using cutting-edge technology to raise awareness, empathy and funds both to respond to humanitarian crises and to bolster support for a new set of sustainable development goals around the globe.
Virtual reality is the ability to really take part in a story that usually you're only a passive spectator on. And you actually get this sensation of feeling like you're being there.
By using multiple cameras that can record in all directions and software that can stitch the images together, virtual reality creates an experience that enables the viewer to see a movie from every angle, above, below, and behind.
It's exciting for the UN, to be involved in some of those early experiments of how we're trying to tell stories, make these films, and work with some of the most cutting-edge people in the industry on it.
So the UN reached out to us and connected, and we realized that there was a great opportunity here to tell some very important stories and to tell them in a way that we thought would be totally new and highly impactful. The way that I define virtual reality at this point in time is basically the hacking of your senses to convince you that you're somewhere other than where you are. Often, I think of it as a sense of vulnerability. So one of the things we've realized in some of our stories is you have a heightened sense of empathy and a heightened sense of connection as a result of that vulnerability.
That heightened sense of connection and empathy was exactly what Gabo Aurora was looking to create at the World Economic Forum in Davos, Switzerland. Not only to inspire leaders to take action, but also to influence donors to increase funding for disaster response efforts.
I'd started experimenting with using innovative ways of advocacy, and I started talking to a lot of different partners and people. What could we do that would be incredible? And someone said, I just came from a meeting at Samsung, and, with some of these other virtual reality headsets, wouldn't it be amazing if you got all of those elite people who could actually go to a refugee camp, or they could go to an Ebola clinic? I just really felt it would get our issues highlighted.
In addition to the screenings at Davos, virtual reality portals have been set up to view the films at high-level political forums around the world. The virtual reality film on display was Clouds Over Sidra.
Clouds Over Sidra is a short film in virtual reality about a girl named Sidra who lives in Zatari camp, which is a Syrian refugee camp in Jordan. And it is a story about a young girl who is giving you a tour of the camp, of what it's like, what her life is like. When the film debuted in Davos, it was a sensation to everyone we showed it to. They come out of it very deeply moved. I'd say half the people who watch Clouds Over Sidra cry. And then we cut a version for UNICEF, where it's face-to-face fundraisers. The way they do that is usually someone with a clipboard on the street in Europe or in different countries. So they thought, well, what if we got people to experience virtual reality on the street?
Quite sad. They don't have a good environment to stay. We try our best to help them.
Early reporting from UNICEF has shown that when using virtual reality, they've doubled the effectiveness of their fundraising efforts.